अस्सलाम वालेकुम स्टूडेंट्स, होप यू all are fine. स्टूडेंट्स आज हम बायो क्लास 11 चैप्टर नंबर 4 एक्सरसाइज एक्सटेंसिव क्वेश्चन आंसर्स सॉल्व करेंगे. सो लेट्स स्टार्ट. फर्स्ट क्वेश्चन है हमारे पास, व्हाट इज फोटोसिंथेसिस? एक्सप्लेन द रोल ऑफ लाइट इन फोटोसिंथेसिस. Photosynthesis is the intricate process by which green plants, algae, and some bacteria convert light energy from the sun into chemical energy in the form of organic compounds such as glucose. This process is fundamental to life on Earth as it provides energy and organic compounds for growth and development. The role of light in photosynthesis is multifaceted. Energy source. Light is the primary energy source for photosynthesis, providing the energy required to drive the conversion of carbon dioxide and water into glucose and oxygen. Excitation of electrons. Light absorption excites electrons in pigments like chlorophyll, which are then transferred to electron acceptors, initiating the photosynthetic process. ATP and NADPH production. Light energy is used to generate ATP and NADPH, essential energy carriers for the Calvin cycle. Electron transport. Light drives electron transport chains in thallikides, pumping protons across membranes and creating a proton gradient. Proton gradient. The proton gradient is used to produce ATP through chemiosmosis, powering the Calvin cycle. Regulation. Light regulates photosynthetic processes such as stomatal opening, chloroplast movement, and enzyme activation. In summary, light is the driving force behind photosynthesis, providing energy, exciting electrons, generating ATP and NADPH, and regulating the process. Without light, photosynthesis would not occur and life on Earth would be vastly different. Next question is, describe structure of chlorophyll. Chlorophyll is a green pigment found in plants, algae and cyanobacteria that plays a crucial role in photosynthesis. The structure consists of porphyrin ring, a planar ring-shaped molecule composed of four pyrrole rings linked together. Magnesium ion. A central magnesium ion Mg2 plus is coordinated to the porphyrin ring, essential for chlorophyll's light-absorbing properties. Phytol tail. A long hydrophobic chain phytol attached to the porphyrin ring, anchoring chlorophyll to the thallicyte membrane. Chlorine ring. A modified porphyrin ring with a reduced double bond, giving chlorophyll its characteristic green color. Methyl and ethyl groups. Various alkyl groups, methyl and ethyl, attached to the porphyrin ring, influencing chlorophyll's light-absorbing properties. Chlorophyll structure allows it to absorb light energy, blue and red light, transfer energy to other molecules, play a central role in photosynthetic electron transport. There are two main types of chlorophyll. Chlorophyll A absorbs light at 430 nanometer and 660 nanometer. Chlorophyll B absorbs light at 450 nanometer and 640 nanometer. These differences in absorption spectra allow plants to harness a broader range of light energy. Next question is, write a note on the photosynthetic pigment carotene. Carotene is a yellow-orange photosynthetic pigment found in plants, algae, and photosynthetic bacteria. It plays a crucial role in photosynthesis acting as an accessory pigment. Carotene absorbs light energy and transfers it to chlorophyll A, enhancing photosynthetic efficiency. Antioxidant: Carotene protects plants from excessive light energy, reducing oxidative stress and damage to photosynthetic apparatus. Light absorber: Carotene absorbs blue, violet, and red light 450 to 550 nanometer, complementing chlorophyll's absorption spectrum. Energy transfer agent: Carotene transfers energy to chlorophyll A, which then initiates photosynthetic electron transport. Color contributor: Carotene is responsible for the yellow, orange, and brown colors in plants, particularly in leaves, flowers, and fruits. There are two main types of carotene: alpha-carotene, found in higher plants and algae; beta-carotene, found in higher plants, algae, and photosynthetic bacteria. Carotene's role in photosynthesis is essential as it enhances light absorption and energy transfer, protects against excessive light energy, contributes to plant coloration. In summary, carotene is a vital photosynthetic pigment that complements chlorophyll, protects plants from excessive light, and contributes to their color. Next question is, explain the arrangement of photosystems. Photosystems are complex arrangement of photopigments, proteins, and electron carriers embedded in the thalloid membranes of chloroplast. There are two types of photosystems: photosystem 1 and photosystem 2. Photosystem 1. Location: Photosystem 1 is located on the outer surface of thalloid membrane pigments. Photosystem 1 contains chlorophyll A, chlorophyll B, and other accessory pigments. Reaction center: The reaction center of photosystem 1 is called P700, which absorbs light energy at 700 nanometer. Electron transport: Photosystem 1 initiates electron transport, generating high-energy electrons that ultimately produce NADPH. Photosystem 2. Location: Photosystem 2 is located on the inner surface of thalloid membrane pigments. Photosystem 2 contains chlorophyll A, chlorophyll B, and other accessory pigments. Reaction center: The reaction center of photosystem 2 is called P680, which absorbs light energy at 680 nanometer. Electron transport: Photosystem 2 generates high-energy electrons, which are used to produce ATP and oxygen through the water splitting reaction. Arrangement of photosystems. Stacking: Thalloids are stacked like coins, with photosystem 1 and photosystem 2 arranged in a specific pattern. Photosystem 1, photosystem 2 supercomplex. Photosystem 1 and photosystem 2 are connected through a supercomplex, allowing energy transfer between them. Antenna complexes: Light harvesting complexes (LHCs) surround photosystem 1 and photosystem 2, capturing light energy and transferring it to the reaction center. Electron transport chain: The electron transport chain connects photosystem 1 and photosystem 2, facilitating electron flow and generating ATP and NADPH. In summary, photosystems are intricate arrangements of pigments, proteins, and electron carriers that work together to harness light energy and drive photosynthesis. Next question is, describe the role of water in photosynthesis. Water plays a crucial role in photosynthesis, serving as reactant. Water is the source of hydrogen atom, which are used to synthesize glucose and oxygen. Electron donor: Water donates electron to the photosynthetic process, replacing those lost by pigments during light absorption. Proton source: Water provides proton H plus ions for the synthesis of ATP and NADPH. Temperature regulator: Water helps regulate temperature in the leaf, preventing overheating. Solvent: Water dissolves and transports essential nutrients and minerals. stomatal regulator. Water availability influences stomatal opening, controlling gas exchange. In the light-dependent reactions, water is split into oxygen, protons, and electrons for photolysis. Electrons are transferred to electron acceptors, ultimately producing ATP and NADPH. Oxygen is released as a byproduct. In the light-independent reactions, Calvin cycle, water is used to synthesize glucose from carbon dioxide using ATP and NADPH. In summary, water is essential for photosynthesis, providing hydrogen atoms, electrons, protons, and temperature regulation, as well as serving as a solvent and stomatal regulator. Without water, photosynthesis would not occur. Next question is, describe mechanism of photosynthesis. The mechanism of photosynthesis involves two stages, light-dependent reactions and light-independent reactions. Light-dependent reactions. First step is light absorption. Light is absorbed by pigments like chlorophyll and carotenoids in thalloid membrane. Next step is excitation of electrons. Energy from light excites electron in pigments which are then transferred to electron acceptors. Next step is electron transport. Electrons flow through a series of electron carriers, ultimately resulting in formation of proton gradient across thalloid membrane. ATP synthesis. The proton gradient is used to produce ATP through chemiosmosis. Next is water splitting. Water is split into oxygen, proton, and electron photolysis, reduction of NADP+. Electrons ultimately reduce NADP+ to NADPH. Light independent reactions, Calvin cycle. First step is CO2 fixation. Carbon dioxide is fixed into a 3-carbon molecule 3-phosphoglycerate via the enzyme Rubisco. Reduction of 3-phosphoglycerate. 3-phosphoglycerate is reduced to form glyceraldehyde 3-phosphate using ATP and NADPH. Regeneration of RUBP. The 5-carbon sugar ribulose 1,5-bisphosphate RUBP is regenerated. Synthesis of glucose. Glyceraldehyde 3-phosphate molecules are combined to form glucose. Overall equation: 6 carbon dioxide molecules plus 6 water molecules plus light energy gives us C6H12O6, 1 molecule of glucose plus 6 O2, 6 molecules of oxygen. In summary, photosynthesis uses light energy to convert carbon dioxide and water into glucose and oxygen, releasing energy that can be used by the plant or other organisms. Next question is, explain in detail the light-dependent phase of photosynthesis. The light-dependent phase of photosynthesis, also known as Hill reaction, occurs in thalloid membrane of chloroplast and involves the conversion of light energy into chemical energy. This phase can be divided into 4 stages. First stage is light absorption. Light is absorbed by pigments such as chlorophyll A, chlorophyll B, and carotenoids in the thalloid membrane. These pigments have specific absorption spectra, allowing them to absorb light energy at different wavelengths. Stage 2, excitation of electrons. The absorbed light energy excites electrons in the pigments, which are then transferred to electron acceptors. This creates a high energy state, allowing the electrons to flow through a series of electron carriers. Electron transport. The electrons flow through a series of electron carriers in the thalloid membrane, ultimately resulting in the formation of a proton gradient across membrane. This gradient is generated by pumping of protons H plus ions across membrane, creating a concentration gradient. Stage 4 is ATP synthesis and water splitting. The proton gradient is used to produce ATP through chemiosmosis, a process where protons flow back across the membrane, driving the synthesis of ATP. Water is split into oxygen, protons, and electrons, photolysis, releasing oxygen as a byproduct. The electrons ultimately reduce NADP plus to form NADPH. Key components: Thalloid membrane, the site of light-dependent reaction. Pigments absorb light energy and transfer it to electron acceptor, electron carrier, facilitate electron flow and proton pumping, proton gradient, drives ATP synthesis and water splitting. ATP synthase enzyme responsible for ATP synthesis, water splitting complex responsible for photolysis and oxygen release. Overall equation: 12 molecules of water H2O plus 12 NADP plus light energy gives us 6 molecules of oxygen plus 12 NADPH plus 12 H plus ions plus ATP. In summary, light dependent phase of photosynthesis harnesses light energy to generate ATP and NADPH, releasing oxygen as a byproduct. These energy rich molecules are then used in light independent reactions to synthesize glucose. Next question is, explain in detail the light independent phase of photosynthesis. The light independent phase of photosynthesis, also known as Calvin cycle, is a metabolic pathway that converts carbon dioxide into glucose using the energy from ATP and NADPH produced in the light dependent reactions. It occurs in the stroma of chloroplast and consists of three stages: carbon fixation, reduction, and regeneration. First stage is carbon fixation. Carbon dioxide enters the stroma through stomata. The enzyme ribosco-ribulose 1,5-bisphosphate carboxylase oxygenase fixes carbon dioxide onto a 5-carbon sugar ribulase 1,5-bisphosphate IUBP, forming a 6-carbon intermediate. The intermediate is immediately converted into two 3-carbon molecules 3-phosphoglycerate 3-PGA. Stage 2 reduction. 3-PGA is reduced to form glyceraldehyde 3-phosphate G3P using energy from ATP and NADPH. This step is catalyzed by the enzyme phosphoglycerate kinase and glyceraldehyde 3-phosphate dehydrogenase. Two G3P molecules are combined to form one glucose molecule C6H12O6. Next step is regeneration. The 5-carbon sugar IUBP is regenerated from G3P and carbon dioxide. This step is catalyzed by enzyme ribulose 5-phosphate kinase and requires energy from ATP. The regenerated IUBP is ready to fix another CO2 molecule, continuing the cycle. Key enzymes and molecules. Rubisco fixes carbon dioxide onto RUBP. 3-phosphoglycerate kinase reduces 3-PGA to G3P. Glyceraldehyde 3-phosphate dehydrogenase reduces 3-PGA to G3P. Ribulose 5-phosphate kinase regenerates RUBP. ATP and NADPH provide energy for reduction reactions. Overall equation: 6 CO2 plus 9 ATP plus 6 NADPH gives us C6H12O6 glucose plus 6 molecules of water plus 9 ADP plus 6 NADP plus. In summary, the light independent phase of photosynthesis uses energy from ATP and NADPH to convert carbon dioxide into glucose through Calvin cycle, which consists of carbon fixation, reduction, and regeneration stages. Next question is, describe cyclic photophosphorylation. Cyclic photophosphorylation is a light-driven electron transport process in photosynthesis that generates ATP without producing any NADPH or oxygen. It occurs in the thalloid membranes of chloroplast and involves the cyclic flow of electrons through a series of electron carriers. Key steps: light absorption. Light is absorbed by pigments such as chlorophyll A and other accessory pigments. Electron transfer. Energy from light excites electrons which are transferred to electron acceptors. Electron transport. Electrons flow through a series of electron carriers including cytochrome B6F complex and plastocyanine. Proton pumping. Electrons flowing through electron transport chain pump protons H plus ions across thalloid membrane. ATP synthesis. The proton gradient generated by proton pumping drives ATP synthesis through chemoosmosis. Electron return. Electrons ultimately return to the starting point, completing the cycle. Characteristics: cyclic. Electrons flow in a cycle returning to the starting point. Light driven requires light energy to initiate electron transfer. ATP synthesis generates ATP without producing NADPH or oxygen. No reduction of NADP+. NADP+ is not reduced in cyclic photophosphorylation. No oxygen evolution. Oxygen is not produced in cyclic photophosphorylation. Importance: ATP production. Cyclic photophosphorylation contributes to ATP synthesis essential for photosynthetic process. Regulation helps regulate electron transport and ATP synthesis in response to changing light conditions. Adaptation allows plants to adapt to varying light environments and optimize photosynthetic efficiency. In summary, cyclic photophosphorylation is a light-driven electron transport process that generates ATP without producing NADPH or oxygen, playing a crucial role in photosynthetic energy production and regulation. Next question is, describe Calvin cycle. The Calvin cycle, also known as Calvin-Benson cycle, is a metabolic pathway that fixes carbon dioxide into glucose using energy from ATP and NADPH produced in light-dependent reaction of photosynthesis. It occurs in stroma of chloroplast and consists of three stages. First stage is carbon fixation. CO2 enters through the stroma through stomata. The enzyme Rubisco fixes CO2 into a 5-carbon sugar, IUBP, forming a 6-carbon intermediate. The intermediate is immediately converted into two 3-carbon molecules, 3-PGA, stage 2 reduction. 3-PGA is reduced to form G3P using energy from ATP and NADPH. This step is catalyzed by enzyme phosphoglycerate kinase and glyceraldehyde 3-phosphate dehydrogenase. Two G3P molecules are combined to form one glucose molecule, C6H12O6. Stage 3 is regeneration. The 5-carbon sugar IUBP is regenerated from G3P and carbon dioxide. This step is catalyzed by the enzyme ribulose 5-phosphate kinase and requires energy from ATP. The regenerated IUBP is ready to fix another carbon dioxide molecule continuing the cycle. Key enzymes and molecules. Rubisco fixes carbon dioxide onto IUBP. 3-phosphoglycerate kinase reduces 3-PGA to G3P. Glyceraldehyde 3-phosphate dehydrogenase reduces 3-PGA to G3P. Ribulose 5-phosphate kinase regenerates IUBP. ATP and NADPH provide energy for reduction actions. Overall equation: 6 CO2 plus 9 ATP plus 6 NADPH gives us C6H12O6 glucose plus 6H2O plus 9 ADP plus 6 NADP plus. The Calvin cycle is essential for photosynthesis as it fixes carbon dioxide into glucose, providing energy and organic compounds for growth and development. Next question is, describe kinds of cellular respiration. There are three main types of cellular respiration. First one is aerobic respiration, occurs in presence of oxygen, producing energy rich ATP, carbon dioxide and water as byproduct. Glycolysis से हमारे पास क्या बनता है, पायरुवेट, पायरुवेट से सिट्रिक एसिड साइकिल, क्रेब्स साइकिल, देन इलेक्ट्रॉन ट्रांसपोर्ट चेन. नेट जो एटीपी यील्ड होती है वो 36 to 38 एटीपी मॉलिक्यूल्स होता है पर ग्लूकोज मॉलिक्यूल. Next is anaerobic respiration, occurs in absence of oxygen, producing energy rich ATP, lactic acid or ethanol and carbon dioxide as byproduct. इसके जो सबसे पहला स्टेप क्या है, इसका भी ग्लाइकोलाइसिस होगा, उससे पायरुवेट बनेगा, देन लैक्टिक एसिड या इथानॉल बनेगा, फर्मेंटेशन, नेट एटीपी यील्ड. इसको हम फर्मेंटेशन भी कहते हैं एनारोबिक रिस्पायरेशन को. इसमें जो नेट एटीपी यील्ड है वो 2 एटीपी मॉलिक्यूल्स है पर ग्लूकोज मॉलिक्यूल. लैक्टिक एसिड फर्मेंटेशन और 2 to 3 एटीपी मॉलिक्यूल्स पर ग्लूकोज मॉलिक्यूल, इथानॉल फर्मेंटेशन. नेक्स्ट इज microaerobic respiration, occurs in low oxygen environments, producing energy rich ATP, carbon dioxide and water as byproduct. ग्लाइकोलाइसिस होगी, पायरुवेट बनेगा, देन सिट्रिक एसिड साइकिल, क्रेब्स साइकिल होगी, तो देन इलेक्ट्रॉन ट्रांसपोर्ट चेन होगी मॉडिफाइड फॉर लो ऑक्सीजन. और इसमें जो एटीपी यील्ड है, नेट एटीपी यील्ड वो है 4 to 6 एटीपी molecules per glucose molecule. Additionally, there are other types of cellular respiration including fermentation, anaerobic process that converts glucose into lactic acid or ethanol, producing 2 ATP molecules per glucose molecule. Glycolysis, first step of cellular respiration occurring in cytosol, producing 2 ATP molecules per glucose molecule. Beta oxidation, breakdown of fatty acids into acetyl-CoA, producing energy-rich ATP and NADH. Each type of cellular respiration has its own unique characteristics, advantages and disadvantages, allowing cells to adapt to various environments and energy demands. Next question is, give an account of glycolysis. Glycolysis is the first stage of cellular respiration, a metabolic pathway that converts glucose, a 6-carbon sugar, into pyruvate, a 3-carbon molecule, generating energy in the form of ATP and NADH. Location: glycolysis occurs in cytosol of cell. Steps: glucose phosphorylation. Glucose is converted into glucose 6-phosphate using 1 ATP molecule. Phosphoglucose isomerase G6P is converted into fructose 6-phosphate. Next step is aldolase. F6P is converted into fructose 1,6-bisphosphate F16BP. Triphosphates phosphate isomerase. F16BP is converted into glyceraldehyde 3-phosphate G3P and dihydroxyacetone phosphate DHAP. Glyceraldehyde 3-phosphate dehydrogenase. G3P is converted into 1,3-bisphosphoglycerate, generating 1 NADH molecule. Phosphoglycerate kinase 1,3-BPG is converted into 3-phosphoglycerate, generating 1 ATP molecule. Phosphoglycerate mutase 3-PG is converted into 2-phosphoglycerate 2-PG. Enolase 2-PG is converted into enol pyruvate ENO. Pyruvate kinase ENO is converted into pyruvate PYR, generating 1 ATP molecule. Net products: pyruvate 2 ATP molecules, net gain 2 NADH molecules. Importance: glycolysis is the primary source of energy for cells under anaerobic conditions. It provides energy for cells under anaerobic conditions when the citric acid cycle and electron transport chain are impaired. Glycolysis is a critical step in the metabolism of glucose, leading to production of ATP, NADH, and pyruvate. Regulation: glycolysis is regulated by feedback inhibition, allosteric control, and substrate availability. Key regulatory enzymes include hexokinase, phosphofructokinase, and pyruvate kinase. In summary, glycolysis is a crucial metabolic pathway that converts glucose into pyruvate, generating energy in the form of ATP and NADH. Its regulation and importance vary depending on cellular conditions and energy demands. Next question is question number 13. Explain oxidation of pyruvate. The oxidation of pyruvate is a critical step in cellular respiration, making transition from glycolysis to citric acid cycle. It occurs in mitochondria, involves conversion of pyruvate into acetyl-CoA, producing NADH and carbon dioxide as byproduct. Steps: pyruvate transport. Pyruvate is transported from cytosol into mitochondria via the pyruvate transporter. Pyruvate dehydrogenase complex. Pyruvate is converted into acetyl-CoA by the PDH complex, a group of enzymes located in mitochondrial matrix. Decarboxylation. Pyruvate is decarboxylated, releasing carbon dioxide. Oxidation. The remaining 2-carbon molecule is oxidized, forming acetyl-CoA. Induction of NAD+. NAD+ is reduced to NADH, generating 1 NADH molecule. Net products: acetyl-CoA, NADH, CO2. Importance: the oxidation of pyruvate links glycolysis to citric acid cycle, allowing for complete breakdown of glucose. Acetyl-CoA enters citric acid cycle, producing more ATP, NADH, and FADH2. NADH generated in this step contributes to electron transport chain, producing additional ATP. Regulation: the pyruvate dehydrogenase complex is regulated by feedback inhibition, allosteric control, and covalent modification. Key regulatory factors include ATP, NADH, and acetyl-CoA levels. In summary, the oxidation of pyruvate is a crucial step in cellular respiration, converting pyruvate into acetyl-CoA, NADH, and carbon dioxide. This process links glycolysis to citric acid cycle and electron transport chain, ultimately contributing to ATP production. Next question is, explain Krebs cycle. The Krebs cycle, also known as citric acid cycle or tricarboxylic acid cycle, is a key metabolic pathway that generates energy through breakdown of acetyl-CoA, a molecule produced from oxidation of pyruvate. Location: mitochondrial matrix. Steps: citrate synthesis. Acetyl-CoA combines with oxaloacetate to form citrate. Isomerization: citrate is converted to isocitrate. Oxidation: isocitrate is oxidized, forming alpha-ketoglutarate and generating NADH. Decarboxylation: alpha-ketoglutarate is decarboxylated, forming succinyl-CoA and generating NADH and CO2. Succinyl-CoA conversion: succinyl-CoA is converted into succinate, generating GTP similar to ATP. Oxidation: succinate is oxidized, forming fumarate and generating FADH2. Hydration: fumarate is hydrated, forming malate. Oxidation: malate is oxidized, forming oxaloacetate and generating NADH. Net products: 2 ATP or GTP, 6 NADH, 2 FADH2, 2 CO2. Importance: the Krebs cycle is a critical step in cellular respiration, generating energy and producing key intermediates. It links glycolysis and electron transport chain, contributing to ATP production. The Krebs cycle also provides building block for amino acid synthesis and other metabolic pathways. Regulation: the Krebs cycle is regulated by feedback inhibition, allosteric control, and substrate availability. Key regulatory factors include ATP, NADH, and succinyl CoA levels. In summary, the Krebs cycle is a vital metabolic pathway that generates energy and produces key intermediate through the breakdown of acetyl-CoA. Its regulation and importance vary depending on cellular conditions and energy demands. Next question is, explain electron transport chain. The electron transport chain (ETC) is a series of protein complexes located in mitochondrial inner membrane responsible for generating energy in the form of ATP through transfer of electrons. Steps: electron transfer. Electrons from NADH and FADH2 are passed through a series of electron carriers. Proton pumping. As electrons flow through the transport chain, protons H plus ions are pumped across the membrane. Proton gradient. A proton gradient is established with a higher concentration of protons outside the membrane. ATP synthesis. Protons flow back across the membrane, driving the synthesis of ATP through the process of chemiosmosis. Components: complex 1, NADH dehydrogenase, receives electron from NADH. Complex 2, succinate dehydrogenase, receives electrons from FADH2. Complex 3, cytochrome BC1 complex, electron transfer and proton pumping. Complex 4, cytochrome oxidase, electron transfer and proton pumping. ATP synthase generates ATP through chemiosmosis. Net products: 32 to 34 ATP molecules per glucose molecule. Water. Importance: the electron transport chain is primary mechanism of ATP production in cellular respiration. It generates majority of ATP molecules during oxidative phosphorylation. The electron transport chain is a critical step in energy production, supporting cellular functions and activities. Regulation: the electron transport chain is regulated by feedback inhibition, allosteric control, and substrate availability. Key regulatory factors include ATP, NADH, and proton gradient level. In summary, the electron transport chain is a critical metabolic pathway that generates energy in the form of ATP through the transfer of electron and proton pumping. Its regulation and importance vary depending on cellular conditions and energy demands. Next question is question number 16. Explain chemiosmosis and oxidative phosphorylation. Chemiosmosis and oxidative phosphorylation are two closely related processes that occur in the mitochondria during cellular respiration. First is chemiosmosis. Chemiosmosis is the process by which energy from proton gradient across the mitochondrial inner membrane is used to drive the synthesis of ATP. This occurs through movement of protons back across the membrane, which drives the rotation of Stark subunits of the ATP synthase enzyme. ultimately leading to oxidation of production of ATP. Oxidative phosphorylation. Oxidative phosphorylation is the process by which energy from the transfer of electrons during the electron transport chain is used to pump protons across the mitochondrial inner membrane, creating a proton gradient. This gradient is then used to drive the synthesis of ATP through chemiosmosis. Key steps: electron transport chain. Electrons are passed through a series of electron carriers pumping protons across the membrane. Proton gradient. A proton gradient is established with a higher concentration of protons outside the membrane. Chemiosmosis. Protons flow back across the membrane, driving the synthesis of ATP through rotation of ATP synthase. ATP synthesis. ATP is produced from ADP and inorganic phosphate. Net products: ADP 32 to 34 molecules per glucose molecule. Water. Importance: oxidative phosphorylation is primary mechanism of ATP production in cellular respiration. It generates majority of ATP molecules during cellular respiration. Chemiosmosis is critical step in oxidative phosphorylation, allowing for synthesis of ATP. Regulation: oxidative phosphorylation is regulated by feedback inhibition, allosteric control, and substrate availability. Key regulatory factors include ATP, NADH, and proton gradient levels. In summary, chemiosmosis and oxidative phosphorylation are two interconnected processes that work together to generate ATP during cellular respiration. Chemiosmosis uses the proton gradient to drive ATP synthesis, while oxidative phosphorylation establishes the proton gradient through electron transport. Next question is, describe substrate level phosphorylation. Substrate level phosphorylation is a type of phosphorylation that occurs during glycolysis and Krebs cycle, where energy is generated through direct transfer of phosphate group from a high energy substrate to ADP producing ATP. Key steps: glycolysis. During conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, a phosphate group is transferred to ADP producing ATP. Krebs cycle. During the conversion of succinyl-CoA to succinate, a phosphate group is transferred to GDP producing GTP, which is then converted to ATP. Characteristics: direct transfer. Phosphate groups are directly transferred from high energy substrate to ATP or GDP. No electron transport. Substrate level phosphorylation does not involve the electron transport chain. Limited ATP production. Substrate level phosphorylation produces a limited amount of ATP compared to oxidative phosphorylation. Importance: supplemental energy. Substrate level phosphorylation provides additional energy for the cell. Anaerobic conditions. Substrate level phosphorylation occurs in the absence of oxygen, allowing for energy production during anaerobic conditions. Examples: phosphoglycerate kinase glycolysis, succinyl-CoA synthetase, Krebs cycle. In summary, phosphorylation that occurs during glycolysis and Krebs cycle, generating energy through direct transfer of phosphate group from high energy substrate to ADP or GDP producing ATP or GTP. So students, हमारे ये क्वेश्चंस कंप्लीट हो चुके हैं. होपफुली आपको समझ आ गए होंगे. If you have any doubt, you can ask in comment section. Please like, share and subscribe my YouTube channel. Thank you.